Professor Gregory is a native of Pittsburgh, Pennsylvania, who received his BS in physics from Georgetown University in 1961. And he did his PhD in physics from MIT in 1966. He had faculty positions at Georgetown University, the University of Tübingen in Germany, as an Alexander von Humboldt U.S. Senior Scientist Fellow, and a position at Clarkson University. He went over to the dark side and held joint faculty and dean positions in the College of Science and Engineering of Gannon University, Leonard Nelson College of Engineering at the West Virginia University Institute of Technology, and Senior Associate Dean of the College of Engineering and Mineral Science, West Virginia University. In addition, he worked in the industry for Westinghouse Incorporation, Raytheon, and Wandel und Goltermann in Germany, and 6 other startup high-tech companies. In 2000, he was appointed as the Dean of the College of Engineering and Applied Science here at UW-M. Later, he stepped down as Dean of College of Engineering in 2005, but he remained on the faculty in Electrical Engineering Department, along with a joint appointment as a professor in the College of Health Sciences, until he retired from UW-M in 2010. He was a co-founder of NovaScan. Now it's called NovaScan Incorporation in 2003. And he is currently the Chief Science Officer and Board Chair of this company. His lifelong research interests have been in the electrical properties of matter, more recently in the electrical properties of the human tissue. Please join me and welcome Dr. Gregory.
Thank you. It's great to be back. I'd like to start off by introducing some of the people who are here. I don't know if you're going to be able to get the camera on them, but who are people who participated in this adventure. Dr. Wendy Mickelson, breast surgeon at St. Luke's. Dr. Ahmed Dahmer, who's also there. Dr. Jim Marks, who's retired from, he's a biologist, but I forgave him for that long ago. Let's see, Brittany Last. I was thinking of your first name, which was Domingo. And Kurt Waldhuder, who's a vice president for the Research Institute at Aurora. And last but not least, wait a minute, I can't remember this guy. Craig Davis, he's our CEO. Okay, so I have a handout that those of you who are here can get, and you'll get it off the screen. It's the end slide that has a QR code and also a URL code that allows you to pick up all the papers that I'm going to be talking about. So I'll have that available for you. The other thing I have, if you want to take a look at it here, is that at the end, I'll be talking about this particular device, which is a way of almost microscopically examining tissue. And the 2 papers I'm talking about are here. So we can, you'll get those. You can get those from the link. So let's get started. The thing I'm going to talk about is the use of this property called charge relaxation as a measure of the rate at which cells transform from normal to cancer. It's a long-term process. Excuse me. And you probably what we're going to talk about. Well, the first thing I'll probably have to do is explain to you what I mean by charge relaxation. And come on in, gang. And the other is that our data, the next thing we'll talk about is the data that we have used that uses charge relaxation as a tool to study cancer. And then data from others that are complementary to ours. And then try to bring that all together to a working hypothesis. And then after that, I will talk about some of our retrospective results of a study that we did this. Hi, Devendra. So let's begin. The whole process is, in this case, is a matter of measuring the impedance of a tissue, which is I treat as a collection of cells. You can pass current through the tissue, and it's either going to go around or through the cell. If it goes through the cell, then you have a cell membrane in the way. And that behaves like an electrical capacitor. You bring up positive charges on the outside, for example, you get negative on the inside. But it's not a perfect capacitor. It's leaky, as we like to say in the engineering profession. It has resistive paths, and in this case, actually paths to take channels of bring sodium ions in and out. So it can be, however, simplified to this following electrical circuit, which is essentially a single path to get to the cell, and then one that either goes through the membrane and is leaky, or it goes around, or it can go around, and that's not so interesting, so I haven't included it in this picture. Now, the reason that this is interesting is that this, if you get to the right frequency range, the characteristic time it takes for the charges to relax, and that's what relaxation means, to relax or to redistribute, so that it is a characteristic time, and it's measured by the resistance, the internal resistance times the capacitance of the membrane. So it's a way of probing the cell membrane. There is then the simple circuit, which is the parallel RC circuit, which was converted by the addition of one single parameter to a thing called the Cole function. That parameter is this thing here. It's a power law. And for those of you who are watching this, who are, how many of you are either biologists or physicians? Yeah. Okay. I have a thing to tell you that you're going to be disappointed in, but probably when you're in grade school, maybe in high school, your teacher told you you're not allowed to take the square root of -1. She lied to you. It's very useful, but I'm not going to expect you to solve that thing. I'll show you what you get. What you get is two parts. This is called the resistive part, and anything time that has square root of -1 in front of it is called the reactive part. And what you have is then the parameter alpha simply is a broadening thing. It broadens the peak in this case and the roll-off. So you get a roll-off, and you get a peak, and you get some spread, which really is because this parameter, the frequency divided by the relaxation frequency, can be different from different parts of the tissue. And the reason that we're going to use this is because from our studies we have determined this is a very useful tool to study cancer. This is some data in this paper that of the two that we're going to, one of my kids probably. And I will turn that sucker off. Okay, thank you, sorry about that. Let me get back to this. What you notice here is this is normal tissue, this is cancerous. The roll-off and the peak are there, but the dots, the red dots, are the fit of the data to the data, which is the solid line. It's hard to see in this screen, and we'll see it on your computer screen quite well, and in the paper you can see it quite well. And that correlation between those is like 3 or 4 nines up here, and 4 going towards 5 nines of the correlation, the Pearson correlation coefficient. It's a very, very accurate thing. And the parameter that we're looking for is this, the one that causes this peak, but it's the parameter that fits to the entire curve. And the second thing that should come out of this paper, if you're going to read it, is that if you have several different very closely spaced curves, because this is as accurate as it is, you can actually subtract out, you can fit a little bit to this peak, subtract out, and you get a second peak. And then you could take the two of them and go through a process. It's called, it's a nonlinear fitting process. And we use the one by a fellow by the name of Powell. And you can get quite a bit more data. So what do you get then when you do all of this? All right, this is a plot of the likelihood that it's cancer in red versus benign, or benign versus cancer in blue. And this is the frequency along this axis is the measuring frequency. And when that equals about 1 e to the fifth, somewhere around 2 times e to the sixth, it's more likely cancer. And if it's on the other side, if it's more likely benign, the peak is the most of the data then fit at a couple kilohertz. Well, the reason this is interesting is this is a logarithmic plot. This is Craig Davis is the one who pointed this out to me. If this was a linear plot, and this is 1,000 times different, this is probably about 1 meter here, maybe on the screen. That would mean we would be 1,000 meters out in the hallway or out in the street to find the other peak. So the two species of tissue have a very strong difference with this parameter. So our published statistics on this, we did 2 papers. Dr. Marks, Dr. Mickelson, myself, my son Chris, and Dr. Tijo and John Schell, John's here around today, but he's working on, we have him working. And in the first paper we published in 2012, out of about 232 cancer specimens and 144, we call them ductal because the cancer is more present in the highly ductal material. Generally, you would use the term benign. And we had 100% sensitivity and about 85% specificity. John Schell, whose background is different than mine, John is more into the data processing, applied the data, and that's the second paper here, which was published last fall, and we have a third that was invited to extend the calculations a bit. But basically what he showed was that the optimum case, you could separate benign and cancer 100% each. So it's, now as you get more and more data, that might fall apart a little bit, but it's pretty good. That was about 180 normal cases and 180 benign or cancer cases. Okay, so the only way we could think of coming up with an answer to what's going on here was to start looking in the literature and see what others have done that are complementary to our work. We couldn't find any data on this number of cancer breast studies in the literature from the actual tissue. But we could find some studies with breast cell lines. Now these cell lines are designated, these are the designators, but there are 4 of them. There's benign, early stage, advanced stage, and what was thought to be metastatic breast cancer, it turns out with more modern techniques they found out that was actually metastatic melanoma, and also this benign case sometimes contaminated. But in fact, it didn't, in this case, the studies that we're going to report, there's 2 of them here. I wanted to point out that it's kind of awkward to have to keep talking about something that's in the range of 10 to the 5th hertz or a little bit bigger. So if you just divide by that number, make a dimensionless number, you have something I call the electrical transformation age. That's my invention, and if it's wrong, it's wrong. But I'll use that as a shorthand. And the 2 studies that we're going to talk about involving these cell lines, first of all, from cells in suspension, and that's different than the Cole thing. So we use the term relaxation frequency, but not Cole relaxation frequency, just to indicate where it came from. And the takeaway is that as the cells transform, the frequency goes up, so that's why I call it a transformation aging. The second study was done the first one was done by a group in London, England. The second at Georgia Tech. And this is really kind of clever. They take single cells, they trap them between two electrodes, a single cell at a time, and they can measure the electrical properties. The takeaway from that is the polarizability of the cell decreases as the electrical parameter, the ETA, increases. Now, I'll show you some. The data I just gave you before for the cell suspensions is here. The data for the cells trapped with a whole cell measurement, they call it, is here. And what they plot here on this side is the magnitude of the impedance versus the frequency, but they do it in the log plot. And if you have a straight line in a log plot, that corresponds to essentially a power law. And this is a very strong power law dependence on frequency here, which gets smaller and smaller as you go through those cell lines. By the time you get to the metastatic case, it's almost zip. Now, the reason for that is that the cells in this two electrode measurement, these cells are not polarizing very well, and they polarize very well in the normal case. So this is yet another piece of data from the literature. The group at Northwestern University, and I had a large NSF SBIR grant, and my program director showed me this work, and he said, I think you're doing the same thing, and I think he's right. What they did is they scattered light from the cells, from an organ, and they found that they could measure the disorder of the cell using this light scattering technique. And this, call your attention to this curve right here. This is a measure of the disorder in the vertical column, and then three different cases: in normal tissue, tumor, and 4 centimeters away, still a lot of disorder. And the implication here is that the cells as a group have changed. That's called field carcinogenesis. And the results, and I just quoted this from the paper, showed that for 3 different cell types, colon, pancreatic, and lung, the cell nanoarchitecture had changed dramatically and over essentially the whole organ. For those who would like to get into the whole business of there's this review article which was excellent, dealing with the use of field characterization. They have studied this in this article for essentially just about every possible use you can think of. The whole idea started back in 1953 with Slaughter. He said that the cancer doesn't arise just from a single cell, but as an anaplastic, which comes from the Greek meaning going backwards, a tendency involving many cells at once. Some of the people I'm working with have also said it may be measuring for the medical and biology people a mitotic index, because what happens is the cells change. Cancer cells tend to want to divide before they really should, and you get a real mess. And the interesting thing is that we're now doing some work in skin, and by the way, again in this paper, they point out that this is probably one of the better places to go to look at field cancernization, because you have access to all the stuff in the neighborhood. And they also point out there's probably a lot of data that's going to have to be changed as time goes on, because you can't trust the fact that you're 4 centimeters away and that it's not a transformed cell. So bring all this together and put it into a working hypothesis. We're going to hypothesize that it's a known long-term process of cells transforming from benign to cancer. that the relaxation frequency has the following connection. First, that the process produces disordered cellular material that causes reduced electrical polarization. That's from the data that I just showed you. That this is a pretty simple thing to be able to prove that as the polarization decreases, the cell's capacitance will decrease, and therefore the peak frequency tends to go up, and that is easy to calculate. That's a back of the envelope. I'm a physicist. The back of the envelope calculation for that. That this may be be a universal property of the cell. Now, I say it may well be because this is the part where we've done it for, and if I can be so bold as to say, I think the breast cancer fits into this model, as did apparently 3 other organs. So in the skin, it looks like it's fitting. So we have now, from our work and others, we have, I think, about 5 organs where this would be a true statement. And then the probable outcomes and the risk of metastasis can be predicted, I'm going to assert, as an hypothesis from the original value of the F relaxation at the time of the surgery, when the patient was originally diagnosed and treated. Okay, so now let's take a test using our retrospective data. Brittany Last, who I mentioned and introduced, and Dr. Mickelson and I have been working on, Brittany's been collecting this data from the Aurora Healthcare Cancer Registry and looking at the data over about a decade, all taken with the same apparatus, and it was an off-the-shelf, well-established piece of equipment, it's what they call a Solitron impedance bridge. I decided to select just those data because then you don't get into an argument as well. Your equipment's different from somebody else's equipment. And so, and we have a lot of data on various things here. Again, you're going to be able to see all of this if you click on the links later. Now, we had 2 things to worry about. One is whether the patient had a recurrence. All the patients that I'm going to talk about right now had cancer. That's not the issue. The cancer is whether it recurred or didn't recur. If you just look at the known time alive, which is one of the things we matched, versus this parameter, which is the dimensionless parameter for the relaxation frequency, it goes from about 4.5 to 7.5 for those that both recurred and didn't recur. The error bars here are standard error. And you notice that by the time you had maybe 8 years worth of data to begin with, but for the lower values. So obviously there is a trend here. So now the next thing you really have to do is go and say, okay, can we separate out the recurrent and non-recurrent cases? And here's one example. What we did is, and this is how we got the likelihood curves previously. We did a histogram of the number of cases of each type, normalized to 100% total each. And we then could take a look at the correlation between different regions here. And if you just eyeball it, you'll see that up until about 12 or so, somewhere around 12, this thing starts to really get quite different between the two. And if you go and look at the correlation coefficient between those, the so-called non-recurrent and recurred cases, you're going to find it's very high, about 0.812. But above that, it's very low. So there's not a strong correlation there. So then you can go and you can do a, you can do the likelihood curves. And again, as I just showed you here, the likelihood is pretty close to 1. Here, it differs between the parameter of 12 and about 15. And then on this part, which is from the original curve, is the downward portion of the curve. It is actually more likely that it would be that it would be normal, or excuse me, non-recurring. Still had cancer, but it didn't recur. So it gives us some hint as to what region would be most important, and obviously, from the point of view of recurrence, this region would be. Okay, so how about the survival of these patients? And this I find a little sad to even have to talk about. But you can take a look at the patients that recur, and you can plot two things. Essentially the average value of the percentage of the patients remaining alive, or on this axis, the length of time that they're alive. And the two curves tend to track. The marked cases, I'll put this up a second time now, of the benign and so on, the various cell lines. And again, the marks here occur basically towards the end point of each. So the A is the benign. It jumps after that very rapidly, and then it starts to go down again at this point, which is the early stage and advanced stage. And even though this is not breast cancer, a metastatic thing, when you get up here around this point, this is about the point where you have to worry about metastasis. So it does seem to hang together. This is something that Dr. Mickelson and I have just started to look at. The staging, you know, patients come in, somebody says Aunt Minnie is stage 1 breast cancer, or stage 2 or stage 3. This is all the patients that had metastatic disease, except for this one case here, which is a little bit below the region we found important from about 4 up. All of the so-called relaxation frequencies are rather high. And on the other hand, the staging, there's a lot of 1s and 2s there. You should probably be 3s. Is that good? So anyway, we're still looking at that. By the way, the reason I use the term working hypothesis, because we're still working on it, and we're hoping we can get some feedback from everybody who attends. I'm not going to go into this at all. This Dr. Rochelle looked into how did the adjuvant treatment compare to the cold relaxation frequency, and basically the same thing. It just simply said when you get above about 4 times 10 to the 5th, that you have a more probability of metastasis. This could be useful for people, Dr. Mickelson, if we get into it in more detail, to determine which treatments are the best. So let's move on now to make one more check mark in the box on types of tissue that can be studied, and that would be with skin cancer, non-melanoma skin cancer. Non-melanoma skin cancer is usually treated with what they call Mohs. Dr. Mohs actually passed away a few years ago, but he was from Wisconsin. He was from University of Wisconsin-Madison. And this procedure involves taking very teeny amounts of tissue. I'll show you an example of a piece of tissue. This is 1 centimeter here. These are each electrodes that are half a millimeter wide and half a millimeter spacing. And what they currently do is each time they take a piece of tissue like this, they make a frozen section slide of it. And the frozen section is then examined under the microscope. Well, that takes a lot of time. And so what we're doing there is trying to speed up the process. In the breast case, we call that being able to get a gross margin. There is no way to get a gross margin for this stuff, so this would be a gross margining technique. And what we do is we change the electrodes that we look at, so we can actually make a map going down here. And let me see if I can turn this damn thing off. So what kind of results do we get with this? Well, it's still very good. Basically, the overall accuracy was about, with both basal cell and squamous cell cancer, were about 98%, excuse me. That was about 98% for BCC, basal cell, and about 100%, but a small number though here. So this is still in the process, but it gives us confidence that the model is still a valid model even with skin cancer. Okay, and the way this is going now, we now have developed this is the actual device. It hooks to a laptop. It has this electrode pattern here. Here we have 24 electrodes. We're eventually going to have something like 96 electrodes here. And we can map by moving or changing the electrode pattern. That has turned out to be a little bit difficult because of the small amounts of impedance that you're dealing with. But the idea then would be to, this is what you would get for a 4-lead measurement. This is a calculation that indicates what region is being sampled. And along the y-axis, the vertical axis, it's rather narrow, but along this axis it's broad. So the red region indicates where we could see, where we could find the cancer. So we can actually pin down where you see it by just switching the electrodes. And then if you switch them, if you combine two, you can do it down essentially the whole thing in the middle. So you can get from this kind of method, you can get, let's see, about 9, 18, 27. And in fact, we can actually pick up these other two regions too. There's another trick we can do. So we can get to about 36, a map of 36 regions. Given that we're talking about spacings here of 0.5 millimeter, that's pretty good. And that's what's needed to make this go. Okay, I'm done. That's the QR code you can use. And if you want to pick up one of these things, you can. Don't pick up my box. But any questions? No questions. Yes.
So, I'm wondering, so can you tell us the device used in clinics and in vivo stages? And those electrodes, so are you doing only on, for example, tissue excise on tissue level?
Well, actually, we have done 2 other studies, and a lot of this is driven by the market to which you are going to try to get the permission to sell. There are 6.2 million cases of non-melanoma skin cancer. There's, what, 250,000 cases of breast cancer per year? In the U.S., yeah. And I'm talking about just the U.S. too with the 6.2 million. So the economics, unfortunately, favor doing it with skin. It has another, if I make a mistake in Dr. Mickelson's case, and she's buying into my thing, that patient could die if she was foolish enough, and she's not, to accept what I say. But as a grossing process, it's probably okay. But we also did 2 other things. We did an electrical mammogram, and Dr. Marks was one of the people who participated in that, and also Dr. Mickelson. And that worked pretty well. But it is not possible, except for very strong cases of cancer, to use it for an electrical mammogram per se. There is a version where you can show that the cancer is there, but to try to get these peak frequencies, the system wasn't designed for that. So we had some trouble getting it published, and we just want to get away from it. However, that I think is a doable. And in fact, we're talking to some, I can't say it because of non-disclosure agreements, but we're talking to some very large companies that are interested in that. The other thing that we looked at is, and with Dr. Mickelson, is to go into the wound with a wand type device. And maybe some of you saw this thing in the newspapers recently about a wand type device that uses mass spectroscopy, which again would have its own difficulties, I'm sure. But we had a lot of trouble making it work, and we finally figured out that there's a lot of wires connected to a patient, and we didn't ask the permission to disconnect them during the study, so we just stopped it. So I think that the answer is the easiest thing for us to do is excised tissue. But we also we have shown we can find the cancer in the breast with an electrical mammogram. We just don't have the money to go and put that into, but we are talking to some companies, and I think eventually that may happen.
So with that, what was the nodule size of the cancer detected actually? Because you have to compete with MRI, which is around 1 millimeter.
Yeah, we're talking about, well, if you're talking the excised tissue, or you're talking about we were, I think the smallest, do you remember, Jim, the smallest one was?
I'm having a hard time hearing.
Oh, the question was, what is the smallest tumor that we were able to find using the electrical mammogram? I think it was below 1 millimeter. Yeah, about 0.5 millimeter.
We do it during the process of grossing the specimen, it's called. There's a technician which will dissect the specimen, expose a section of it, and usually with the electrode that we had, a 2 millimeter, 3 millimeter section is detectable, although there's a lot of interference from the adjacent tissue hit. But generally, anything that is about 3/4 of a centimeter.
Certainly see it. Yeah. It actually with the one of the reasons we went to doing the skin cancer is we were able to develop this newer electrode pattern, and we can actually map that now. You're getting down to below 1 millimeter. Now, in the case of the imaging, the interesting thing is the further away from the tumor, it's like a light coming through the tunnel. You can see a little point coming through, and all of a sudden you realize that's a train. You better get off the track. So we can get. We can actually go sub-millimeter in the size, because once it gets very close to the surface, and it's a question of how close, we can see a depth of maybe 4 centimeters. When you compress the breast, that's about what you get, and we can get it from either side. So we can actually we could double that if there's no particular need, and that's what I think would work. But we're not pursuing it actively right now, simply for economic reasons. Sorry.
Any other questions? Okay. We thank our speaker.
Thank you.